Everyone, my name is Miriam. I'm a rising junior in chemical engineering at NC State, and the research I conducted was to study Alzheimer's disease using C. elegans as a model organism. So Alzheimer's disease has many characteristics, but one of the main physical changes that happens in the brains of patients is the accumulation and aggregation of two proteins, the amyloid beta protein, which was the focus of my research, and this protein forms plaques around the neurons as well as in the synapses of neurons, and the tau protein, which tangles with neurons. So the goal of the project was to express amyloid beta in C. elegans and see how this protein interacts with specific neuron types and how it progresses Alzheimer's disease. So C. elegans, there's a picture of it in the poster. It's a very simple nematode, but it has similar molecular and biological pathways as humans, which makes it very ideal for research purposes. So how exactly is amyloid beta produced? In humans, amyloid beta is produced when the amyloid precursor protein, which is a protein embedded in the neuronal membrane, is cut by two enzymes. The beta secretase cuts first, and it leaves the C99 fragment of the APP protein embedded. There's a picture of it in the poster. From there, the gamma secretase makes a second cut, and this second cut releases amyloid beta extracellularly. Worms have a similar protein called the APL protein in their membrane, but this protein is cut by alpha secretase first, followed by gamma secretase, and this protein also lacks the amyloid beta fragment within it. So worms naturally cannot produce amyloid beta by themselves. So to get them to produce the amyloid beta protein, we had to get them to produce the C99 fragment first. And to do that, we use Gibson assembly. So we used the GH8 plasmid and we inserted in the C99 gene into the plasmid. And this process took many weeks because the GH8 plasmid is very large and we had to experiment with different annealing temperatures and extension times and different DNA polymerases. But eventually we did get it, and we sent it off to sequencing to confirm that the C99 gene was oriented and ligated correctly. And once that was confirmed, we started microinjection. And to do that, we used the M-cherry plasmid as well as the GH8 plasmid with the C99 fragment within it. The M-cherry plasmid it produces a protein that will fluorescence under certain lighting, and this just confirms that the worms that we're working with were microinjected correctly. So if you look on the microinjection section of the results and discussion, you will see that some of the worms are lighting up, and these are the worms that we will use to study to make sure that the C99 fragment is being expressed. So in the future, we will create a worm line containing the C99 gene, and then from there we will use real-time PCR and antibody staining to make sure that that protein is being expressed. And once that is confirmed, the worm's natural gamma secretase should be cutting at the correct spot in the fragment and releasing amyloid beta extracellularly, so we can start imaging the worms to see how this protein causes neuronal degradation. Thank you.